So in this session, next speaker is Professor Samir Jadhav. Professor Samir is from IIT Bombay, and his group is working on biomaterials, cell mechanics, simulations of biological systems, and liposomal drug delivery systems. So Professor Samir will be talking more about the simulations and then validation by experimental methods. So over to you, sir.
Yeah, so good morning, everyone. And I'll not just be talking about our recent work on Gala Peptides, but also on other work that we have done in our lab. And I'll start from where we started our journey. Uh, so I'll skip this slide. We're all aware about liposomes as drug carriers and the advantages they provide. So this talk I will be talking about the synthesis of liposomes from giant vesicles. Then some properties and characterizations we have done for first by making these chitosan coated vesicles and PEG-lated vesicles. Then pH modulated poration of vesicles using Gala, and some insights from atomistic simulations, how pH dependent peptides are able to porate these vesicles. Yeah, so typically we see that whenever we want vesicles for delivery, there would be initial coating of lipid particles and then a hydration at a higher shear, or of course there are other methods like emulsion methods which can be used. Problem in most of these cases is that you have these multilamellar vesicles, and if you want unilamellar vesicles, what do you do? So what we have done is we've used electroformation to form these large or the giant unilamellar vesicles, and then we pass them through a track etched membrane, polycarbonate membrane, to give us unilamellar or SUVs, small unilamellar vesicles. The disadvantage typically when we get MLVs is you need several passes, sometimes more than 10 passes through the membrane to get the size you want. Whereas when we did this extrusion of giant vesicles, what we found was we could get the size we needed only by a single pass through the membrane. So that's the setup where you have an electroformation that makes you the giant vesicles. And then we have a filtration unit so that we could get the giant vesicles, and this is a step which may not be needed, but if you want a particular size, this is really something which is important. And then finally extrusion through a track etched polycarbonate membrane. So this is what the membrane looks like. So there are these pores. We also did measure the size of these pores from what was reported by the manufacturer versus what we were able to estimate. And here we see basically the extrusion of MLVs by the traditional method, and then the extrusion of GUVs. As you can see, the extrusion from these MLVs requires a larger pressure. Compare that to the GUVs, we see that the size we need is obtained at much lower pressures. And of course, you can see here the polydispersity index is also much lower when we have these extrusion of giant vesicles. This is the encapsulation efficiency, and we see again that GUVs give a significantly larger encapsulation efficiency. This just shows that a single pass allows us to get the size, because multiple passes there's no further reduction in sizes. And this basically is using different membranes and the extrusion pressure required to get the minimum attainable size of the vesicles. We could then basically model this process where based on the size of the pores in the membrane, one can collapse all the data onto a single curve. And as you can see that towards the lower part here, the size is not matching, but that is because not because of the problem with the model, it is actually related to the problem with the experiments themselves. That's why I said that the initial filtration step, which removes the smaller vesicles, that basically caused this under prediction by our model. But once we removed that, the model fits very well. We also looked at properties of the vesicle using the micropipette aspiration assay. And this assay allows us to, so these are just in terms of showing that chitosan is actually coated onto the vesicles. But how does it affect the mechanical properties? For that, we carry out the micropipette aspiration assay, where we can measure both the bending modulus as well as the area compressibility of the membrane. And as you can see here, at different pH, we get different bending modulus, whereas the area compressibility modulus remains unchanged. We also looked at permeability of the membrane, as well as diffusivity in the membrane using FRAP. Since PEG vesicles are used because of their potential for preventing immune response, we carried out extensive studies on the mechanical properties of vesicles, how the PEG molecule, how the PEG grafted lipids affect these mechanical properties, and we saw that they fit the models well, and we also see how the molecular weight of the PEG affects the properties of the vesicle. That in turn tells you how they susceptible they will be to shear and the like. We also looked at the lysis tension, basically the loading at which these membranes would rupture using our technique. So coming to the pH dependent modulation of vesicles using Gala. Gala is basically this peptide which is 30 amino acid in length, and it has these repeating units of glutamic acid, alanine, leucine, and again alanine. And this is what it looks like, where at different pH it shows at pH 5 and below it is a helix, primarily a helix, whereas at pH 7 it is mostly a random coil. And what happens is this particular peptide is able to assemble into the membrane. Mostly 10 mers are able to form a pore within the membrane. These are giant vesicles, and in the presence of Gala, and you can see here at 40 minutes onwards that the shape of the vesicles is no longer spherical, because poration causes them to lose fluid. And also here you can see that the membrane integrity is affected because of the Gala peptide. So we also measured the kinetics of these pore formation by using a dye which would then get into the membrane if it is porated. And these are studies. The initial is basically just a change in pH does not let the dye in. And when the dye is present, when the peptide is present, you see that at pH 7, the vesicles do not allow the dye in. But as we reduce the pH, we can see that the vesicles now have intake of the dye. We measured the kinetics of this process, and we were able to estimate the kinetics in terms of the rate of increase in the pore density in a membrane, as well as the rate constant for pore formation within the membrane. And this was independent of the vesicle diameter, as can be seen here. Finally, we also looked at this particular peptide in terms of how it behaves, or what are the configurations at different pH, and this is using all atom simulations. So on the left, it is at pH 4 that the peptide is simulated, and you see that it has a helical configuration, whereas at pH 7 it's a random coil. And of course, this also is quantitated by measuring the root mean square deviation from a reference configuration. And as you can see, for pH 4, these values are much lower compared to at pH 7. Same goes for the RMSF, the root mean square fluctuations. On the left is at pH 4. These values are much lower compared to pH 7, where it's a more flexible molecule, and therefore it sees a larger fluctuation. This is the Ramchandran plot of the dihydrals, and typically on the left here is what you see, a right-handed helix, whereas at pH 7 it sees more of beta sheets and a random coil. This at pH 7 we look at basically the how the helical structure, where the first residue is then overlapped by a pitch. After one pitch, the fourth and fifth residues are directly above it, and that is how we see this that in the at pH 7 they are not in contact. So a lighter color shows that they are not in contact. So lesser time in or fraction of time in contact. Whereas in case of pH 4, we see that these are blue or black, which means that these residues are in contact, and therefore shows the integrity of the helical configuration. We also looked at aggregation of the Gala peptide, and we could see that it is it forms aggregates at pH 4, whereas at pH 7, even if it does aggregate, it has very loose aggregates compared to a much tighter aggregation at pH 4 compared to pH 7. And this is the solvent accessible surface area of the peptides. Again at pH 4, it's a very tight curve, whereas at pH 7, because of the high fluctuation and a relatively open structure, it is I mean, we see that very high values of SASA are also accessible. So this is just a broad overview of the work we have done. This work was the initial work related to the extrusion studies was done by Yogita, and the membrane characterization related to mechanical strength were done by Hanipriya as well as Amit. And Somava and Rakesh are the ones who were involved with the simulation studies. And of course, close collaboration with Rochesh, who's here, as well as Rakesh is also here. He's put up a poster, so I would encourage you to take a look at that. And as Mahesh has mentioned, all this work started with the IRFA project anchored by Professor Devang Kakkar. And of course, this nanomission project was in collaboration with ICT. That formed the later part of the studies that we have done. Thank you. So I'm happy to take questions.
Yeah. Thank you, Professor Samir.
Exciting work with the Gala peptide. It's a fusion peptide. I wonder, like, if you can develop similar kind of model for the prediction of ionizable lipids interaction with the endosomal membrane. So that can be a very good tool to screen a lot of lipids that can be used for liposomal delivery.
So yeah, in terms of the model, again, Rakesh and Rochesh, they have looked at a simulation of ionizable peptides and actually using electrical charge in our simulations, both in Atomistic, of course, we can do it easily, but more importantly, we also have in-house DPD code, which you can run on a PC rather than having a large cluster of machines. So in terms of just the ease of simulation, it's a mesoscopic method, dissipative particle dynamics, but it's much faster than atomistic, full-fledged all-atom simulations. Yeah.